Welcome, my dear students. Today, we are going to have a quick revision on the chapter Principles of Inheritance and Variation. Okay. Now, what is the definition of genetics? Genetics, it is the definition. So, it is this heredity, it is the inheritance and variation. Genetics deals with the inheritance and variation. So, which are passed from the parents to progeny. Inheritance means what we inherit from our parents, that is our genetic material, which forms our heredity, and variation is lack of similarity, how or each individual is very different from its parents. That is lack of similarity, that is variation. So genetics is inheritance and variation, or it is the basis of heredity and variation. And these ancients of our ancients before 8000 BC, they knew that something is hidden in sexual reproduction. Something is hidden in sexual reproduction, and these variations are hidden in the sexual reproduction, and they made this and they selected animals which show very good variations, and they artificially made the selection. Artificial selection means the selection made by man, that is artificial selection. So artificial selection and natural selection. Natural selection, nature is selecting which individual is best suited for which character of individual is best suited for the prevailing environmental conditions. That is natural selection. Here, this man is making the selection. According to his will, he is making selection. So by artificial selection, that is the cows showing good variations, and domestication, and they are grown, they are bred, and such like. That is what is called domestication from the ancestral wild cows, from the ancestral wild varieties, and they developed a new variety known as Sahiwal breed of cow. Okay. Now we know that this Mendel is the father of genetics, and he worked for 7 years on Poissum sativum. And that the pea plant, and the main feature of this Poissum sativum is that Poissum sativum is naturally self-pollinated plant. It is naturally self-pollinated plant, but we can also make artificial pollination. So both type of pollination is possible. Naturally self-pollinated, but artificial pollination is also possible. And Mendel was successful because of 3 reasons. Number 1 is he had a very large sampling data, very large number of plants. He did experiments, and this another time statistical and mathematical logic were applied to biological problems. So statistical and mathematical formula were applied to biological problems, and he had a very large sample, and also he continued his experiments for several generations. So these are the 3 main reasons for success of Mendel: statistical, mathematical logic applied to this his studies. Second, he had a very large sampling data. When data is maximum in number, the results will be more accurate. And he continued his crossings, his references, his studies for successive generations. Then Mendel actually conducted artificial or cross-pollination. And he developed true breeding pea lines, true breeding plants. How did he develop true breeding true breeding pea lines? That is by continuous self-pollination. And after continuous self-pollination for several generations, tall plants will produce only tall, dwarf will produce only dwarf. These violet color flowers will produce only violet, white flowers will produce only white, like that. So each character is made pure by continuous cell pollination, and once it is pure line, there is stable trait of inheritance. The character will be inherited for several generations, and Mendel made such 14 true breeding pea plants. 14 true breeding pea plants. Actually, Mendel studied 7 characters and 14 traits. For example, trait character means height of plant. Plant height. Plant height is the character, and it has two traits. That is, one is tall and another is dwarf. So these are the two traits, and this is the plant height is the character, and these two are the traits. So like that, Mendel raised 14 such true breeding plants by continuous cell pollination. Then in the first, then these are the 7 characters, 7 studied by Mendel: stem height, tall, dwarf, flower color, etc. 7 characters and 14 traits, etc. Now in the first cross, Mendel crossed a tall plant, a tall and dwarf plants. Mendel crossed a tall and dwarf plants, and the first to produce the first hybrid generation. And this generation is called filial or first filial or progeny generation 1. And in the F1, Mendel observed that all the F1 plants were tall like the parents, and no dwarf plants was formed. Then that was this first cross. Mendel crossed a tall plant, tall into dwarf cross, and in the F1, all plants were tall. And then after this F1, he allowed these plants to undergo self-pollination, self-fertilization. So these F1 plants are allowed for selfing and to raise the F2. And in the F2, Mendel got tall and dwarf plants in the ratio 3:1. Tall and dwarf plants in the ratio 3:1. So when tall plants, the first cross was between a tall plant and dwarf plant, and Mendel got all the F1 plants were tall. And on selfing the tall plants, Mendel got the F2 generation tall into dwarf, tall is to dwarf in the 3:1 ratio. Then, so these tall plants were just, and dwarf plants were just identical, same thing, just identical to the original plants. Then that means there is no blending. That is the most important thing of crossings of Mendel. There was no blending, mixing of the two characters. So that is why in the F1, both the characters were there, but due to some reasons, only tall appeared and dwarf remained recessive. So dwarf did not appear, but they do not mix together. The genes for tall plant, the alleles for tall plant remained as such, and this dwarf allele also remained as such. They did not mix together. And that is why in the F2, Mendel got these two characters, that is tall, the two traits Mendel obtained, tall into dwarf, tall and dwarf in the 3:1 ratio. Okay. Now, next and most important thing, there was no blending. Then Mendel did same experiment with all the 7 characters, and he found that with all the 7 characters, Mendel did the same experiment. And so he got, when two parents of the contrasting traits are crossed, tall into dwarf, tall into dwarf, pure tall into pure dwarf, and gametes. This was the F1. F1 was tall, heterozygous tall. And this F1 was self. And in the F2, Mendel obtained 3 tall plants is to 1 dwarf plant, 3:1 ratio. And this 3:1 ratio is known as Mendel's monohybrid ratio. And in the 3:1 ratio of the three F2 tall plants, this was pure homozygous tall. and these two were heterozygous tall, and this was pure dwarf plant. So the ratio 1:2:1 is known as genotypic, genes, allelic or factors, how the factors are combined. These are homozygous tall, two are heterozygous tall, and one is homozygous recessive tall. So 3:1 is phenotypic ratio, and 1:2:1 is genotypic ratio. Now, when Mendel conducted experiments with all the other characters, he found that something is being stably passed down from parents to offspring through the gametes. And he called them as factors, stably from parents to progeny, and he called them as factors, and now we call them as genes. And genes are there for the units of inheritance. The characters are inherited through genes, and genes which code for a particular contrasting trait. Particular contrasting traits are known as alleles. For example, tau trait, recessive dwarf trait, etc. They are, on another definition for alleles, they are slightly different forms of the same gene. Okay, now this homozygous. Homozygous means when both the alleles are of same type, it is known as homozygous. Heterozygous means when both the alleles, one is having dominant allele and the other is having recessive allele, is known as heterozygous condition. Genotype means genetic makeup, and genetic makeup, this is homozygous tall, this is heterozygous tall, like that. And phenotype means external appearance, whether it is a homozygous tall or heterozygous tall. Externally, the plant is tall plants. Now, what is meant by this dominant and recessive? When in when two alleles are different alleles are combined in an offspring, capital T and small t, one of the allele dominates over the other, and the allele which dominates is known as dominant, and the allele which is suppressed is known as recessive. But these two alleles remain as such in a particular individual, and there is no blending, and they will separate during gametic formation. Next, this is another method of solving the problem. This is known as Punnett square. In Punnett square, this the production of gametes by parents, formation of zygote, F1 and F2 plants can be understood from a diagram known as Punnett square. This is Punnett square, and this is developed by a British geneticist, Reginald C. Punnett. It is a graphical representation definition. It's a graphical representation to calculate the probability of all possible genotype of the offspring in the genetic cross. That on one side male gamete, on the other side female gamete. This single glance we can get the phenotypic ratio, genotypic ratio, gametes, parents, everything. That is this Punnett square method. Next is this. After studying this, Mendel developed another technique known as test cross. Test cross. Suppose to know the genotype of the F1, F2 parent, whether it is dominant or heterozygous, to know the genotype of the dominant parent, whatever may be the dominant condition, it can either be homozygous or it can be either or heterozygous. And when this is crossed with a recessive genotype, So the recessive genotype, we know the genotype. In the recessive condition, the alleles, both the alleles will be in recessive form, the homozygous form. So the phenotype is itself the genotype. So in all recessive conditions, both the alleles will be in the recessive condition, so that in a recessive and homozygous condition, so that the phenotype is clearly telling us about the genotype. So this we know. Whether the tall plant is homozygous or heterozygous, we do not know. Suppose the given this plant is homozygous, we get after this test cross, we get only dominant character, tall parents, tall character. Suppose after test cross, we get only tall dominant character, then the unknown parent in question is homozygous. Suppose it is heterozygous condition, in 1:1 ratio, we get both dominant and recessive character. This is known as heterozygous condition. And when after a test cross, if we get the progeny 1:1 with dominant and recessive character, then the test cross parent is in heterozygous condition. So that is with regard to this test cross. Then next is, this is another example of a test cross given in your book. This is violet color and white color. Violet is, and this is dominant, and it can be homozygous recessive is white, and this is homozygous recessive white, and this is crossed with the violet flowers, and violet, all the flowers are violet here. So you can solve this problem. Then after working out this cross, monohybrid cross with all the 6 characters, Mendel developed 2 laws known as principles of inheritance. Principles of laws of inheritance, and first law is law of dominance, and second law of segregation. The first law says that characters are controlled by discrete units called factors or alleles. And factors occur in pairs like that, homozygous or heterozygous condition like that. And in dissimilar factor, in the combination of dissimilar factor, one of the factor will dominate over the other factor. The factor which dominates is known as dominant factor, and with the suppressed is known as recessive factor. Okay. Now, second law, most important law of this Mendel is law of segregation. Law of segregation. Law of segregation is also known as law of purity of gametes. Law of purity of gametes. According to those, these alleles do not show any blending, and both the characters are recovered in as such in the F2. Why this both in the F2 generation both the characters are formed? It is due to the non-blending. There is no blending in the F1 condition. F1 was like this, but these 2 alleles, they get separated during gametic formation. Actually, the chromosome carrying capital T is segregating, and chromosome carrying small t is segregating. So during meiosis, this chromosome moves to one gamete, this moves to another gamete, and the gametes contain only one of the alleles. So not the 2 alleles. That is known as law of purity of gametes. And the main reason is there is no blending in the F1 generation. So this law is the only law. Mendel, after is the monohybrid and dihybrid cross, proposed 3 laws of inheritance. And the only law which stands till there is no modification. The only law without modification, even today, it is known as law of segregation or law of purity of gametes, because this is actually meiosis. Mendel did not know what is meiosis. Only after his death, meiosis and mitosis were discovered. But mathematically, he found that only if there is segregation, the next generation can be produced in such a manner. So he found that these gametes, in the alleles or factors of each character, they separate during gametic formation. That is purity of gametes and law of segregation. The chromosomes or alleles are being segregated. And this is the only law which stands even today without any modification. And the cytological proof of this law is meiosis. Next is, actually, this is not a Mendel's work. This is a modification of monohybrid ratio. Monohybrid phenotypic ratio was 3:1 and genotypic ratio was 1:2:1. Immediately, soon after Mendel's death, scientists were crossing all types of plants and animals. And they found that in most of the plants, they did not get the ratios of 3:1 and 1:2:1. And they went to find out the reason why they are not getting the 3:1 and 1:2:1 ratio. And then came across some type of complicated inheritance. Like one of the complex inheritance is interaction of factors. Several interaction of factors. One type is known as incomplete dominance. Incomplete dominance. In incomplete dominance, it was found that sometimes the F1 had a phenotype that did not resemble either of the 2 parents and was in between the 2. The F1 plant in Mendel's cross, the F1 resembled either of the parent. Usually it is the dominant parent. But here, in this particular case, interaction of factors, incomplete dominance, the F1 did not resemble any of the parents, but the character of the F1 was in between the 2 parents. For example, when a red flowered snapdragon plant is crossed with a white flowered the snapdragon plant, the F1 was red in color. And when the F1 was selfed, F1 was selfed in the F2, Mendel obtained. In the F2, Mendel obtained 1:2:1 ratio. This is red, this is pink, and this is white flower. Red, pink, and white in the ratio 1:2:1. Okay, so the phenotypic ratio is red, pink, and white, that is in 1:2:1. At the same time, genotypic ratio is also 1 homozygous red, 2 heterozygous. It cannot be red, it is pink, and this is white. So 1:2:1 genotypic ratio, and this is phenotypic ratio. And in incomplete dominance in this snapdragon, the genotypic ratio is 1:1 and phenotypic ratio is 1:1. And the Mendel's monohybrid phenotypic ratio is modified into 1:2:1 of this incomplete dominance of snapdragon. The reason in Mendel's cross, capital T was completely dominant over small t, whereas here, capital R is not fully dominant over small r. It is only incompletely, partially dominant over small r. Then another modification of Mendel's monohybrid cross is this F1 resemble either of the two parents, that is Mendel's, either of the two parent dominant character, or was in between the two parents, that is incomplete dominance. But in codominance, the F1 resemble both the parents. F1 resemble both the parents. A good example is different types of red blood cells of determined ABO blood group. ABO blood group is actually determined by the gene I. I has 3 allelic forms. It can be IA, it can be IB, or it can be small I. So these are multiple alleles. When more than 2 alleles are present for a character, for a trait, for a character in an organism, it is due to multiple allelic. So ABO blood group system shows a multiple allelic series. And these different blood groups, A group can be homozygous A, or it can be heterozygous A. B group can be homozygous B group, or it can be heterozygous B group. And there is another group, when both these A and B combine, there is AB group and small I, small I. Now, these different blood groups are due to the presence of these different sugars found on this RBC membrane. RBC membrane. IA produces a particular sugar, IB produces another different sugar, and small I produces no sugar at all. And the small I is supposed to be the most recessive of this. Both IA and IB are equally dominant, and they both are dominant over small I. And small I is the most recessive, and when small I come together, we get a O group blood. Okay. Now comparing this, we can see that this blood group system is, ABO blood group system is a multiple allelic series, and IA and IB, AB group is codominant, and small I is most recessive condition. And there are 3 alleles, and there are 3 alleles and 6 genotypes and 4 phenotypes. The different phenotypes are A group, B group, AB group, and small I, O group. So there are 3 alleles, 4 phenotypes, 3 alleles, 4 phenotypes, and 6 genotypes. 6 genotypes like IA, homozygous IA, or heterozygous IA, small I, like that. 6 genotypes and 4 phenotypes and 3 alleles in ABO blood group system. Okay, so far we have been discussing about this one single gene influencing single character. But there are examples where one single gene can influence more than one character. So one gene influencing more than one character, such that that is known as manifold effect of genes. And there are two, one example, beautiful example is given, starch synthesis. Starch synthesis in seeds, in pea seeds, it can be, this seed shape can be capital B, round shape. This is wrinkled shape, and heterozygous is round shape. So both these are dominant recessive. Seed shape shows dominant recessive condition, whereas starch grain size, this shows capital B shows large starch grains, and small b shows small starch grains, and heterozygous condition produces intermediate starch grain. So with regard to starch grain size, the character is incomplete dominance. So one gene affecting more than one character. One is on this dominant recessive seed shape, and the other is starch grain size. Both are, one is dominant recessive and another is starch grain size is this starch grain size is at this incomplete dominance. Then after crossing this the monohybrid cross, Mendel went for to cross Mendel went for to study two characters at a time, and Mendel crossed round yellow plants with the wrinkled green plants, and we know that round is dominant over wrinkled and yellow is dominant over green. And Mendel crossed these round yellow plants with the wrinkled green plants. And these are the gametes, and the F1 Mendel obtained all the plants were round yellow. And when the round yellow plants were allowed for selfing, Mendel obtained this round yellow, 9 round yellow, 3 round green, 3 round wrinkled yellow, and 1 wrinkled green in the ratio 9:3:3:1 ratio. So this is Mendel's dihybrid phenotypic ratio. Dihybrid phenotypic ratio is 9:3:3:1 ratio. And this is phenotypic ratio, and the genotypic ratio is a very big ratio, 1:2:1, 2:4:2, 1:2:1. This is genotypic ratio. And totally in monohybrid cross we got 4 individuals, 3:1, whereas here we get 16 individuals. So monohybrid cross for 4 individuals. For total progeny in the F2, total progeny was 4, and in this dihybrid cross, the total number of progeny in the F2 is 16. So we get 2 parental types. Round yellow is a parental type, and wrinkled green is a parental type, and these two are new types. So the possibility of this, why this, after this dihybrid cross, Mendel proposed one law known as this law of independent assortment. Law of independent assortment was proposed by Mendel. According to this law of independent assortment, this capital Y, yellow seed, this is small y, this is round shape, this is wrinkled shape, round yellow and wrinkled shape. So in this, according to this law of independent assortment, according to Mendel, this Mendel was 100% correct with all his results, but the other plants and animals may be showing different types. It's variations in basically Mendelian variations. So that is Mendel's, this all the cross, all this, these were the types of genes that were situated on 4 different chromosomes. And this Y can combine with capital R, or Y can also combine with the small r. Small y can combine with the capital R, or small y can also combine with the small r. So different combinations are possible. That means when in a dihybrid cross, the law states that when 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. One pair of character color is independent of seed shape. So this capital Y, it is separated on, it is situated on a separate chromosome, Y on another chromosome, R on another chromosome, small r on another chromosome. So each one during meiosis, when meiosis takes place, segregation takes place, and each one can go for different kinds of combinations. That is why Mendel obtained 2 parental types and 2 new types. 2 parental types, round yellow round yellow and wrinkled green. Two parallel types, round yellow and wrinkled green. At the same time, two new types, round green and wrinkled yellow. Two new types also Mendel obtained. So that is Mendel's dihybrid cross. Now, this after this Mendel, now we go for this chromosomal theory of inheritance. This chromosomal theory of inheritance, then in 1908, Mendel's papers were published in 1865, and it remained unrecognized till 1900. The reasons why these papers remained unnoticed, unrecognized is that, first of all, the communication was not easy. Only very few preprints he took. Secondly, the concept of genes, Mendel's words are stable and discrete unit, which is passed on from parents to progeny, and but they did not blend together, and the alleles did not blend together, was not accepted by the contemporaries because most of the characters seen in nature is of this blending type. Most of the characters are seen in nature is blending type. Then third, application of mathematics and biology in mathematics in biological principles. And finally, Mendel suggested that genes or factors were discrete unit, and Mendel could not provide any physical proof that the physical existence of genes. Then in 1900, three scientists from three different countries, Deveries, Cons, and Sharma, rediscovered Mendel's papers. And by that time, this meiosis and mitosis were detailed studied, and staining techniques developed. And in this, in 1902, chromosome during meiosis worked out, and Sutton and Boveri noted that the behavior of chromosome was parallel to the behavior of genes. There is a parallelism between the behavior of chromosomes and behavior of genes, parallelism. And this parallelism is Mendel has already explained in law of segregation, law of purity of gametes, segregation of chromosomes. And this law of segregation is actually proved by this Sutton and Boveri by studying meiosis. And the most important point is two alleles of a gene pair are located on homologous chromosomes on homologous sites. Homologous chromosomes with the homologous sites. The two alleles will be present on homologous chromosomes on homologous sites. So that is this chromosome theory of inheritance. Then this following this idea, explain now this Morgan's work, this Thomas Hunt Morgan, T. Hunt Morgan, the sexual reproduction, how variation is produced during sexual reproduction. And this the first time this Drosophila melanogaster was introduced into genetics by Morgan, and why an organism, why he selected this Drosophila melanogaster. There are three or four reasons. One is it can be easily grown in simple grown can be grown in simple synthetic medium. They complete their life cycle within 2 weeks. Single mating produces a large number of progeny, and there is clear differentiation of sexes, and there is easily detectable variation, hereditary variations are also present. And now this linkage and crossing over, then this Morgan crossed a yellow bodied white yellow bodied white eye color female Drosophila with a brown bodied and red wild type male. And when he crossed, he obtained an F1, and when the F1 was selfed, in the F2, Mendel obtained 98.7 parallel types and only 1.3% of recombinant types. And it is a dihybrid cross, so he was actually expecting a 3:3:1 ratio. Mendel's ratio, but dihybrid cross ratio, but Morgan obtained a ratio far more number of parental types than very little number of, negligent number of these recombinant or new types. Then, whenever he made a conclusion that whenever there is in a cross, whenever there is no dihybrid cross, whenever we did not get this 3:9:3:3:1 ratio, that means there is no independent assortment. There is no independent assortment, or in other ways, only where there is independent assortment, we get only 9:3:3:1 ratio. And the reason for this large number of parental proportion is a phenomenon known as linkage. Linkage means when two or more genes are seen on the same chromosome, they show a tendency to move as a group, and this is known as linkage. And how these two new forms are formed? Two new forms are formed by recombination or crossing over. And another feature is that after studying this cross, Morgan also crossed another dihybrid cross with wild type males. Another type males with white-eyed and miniature wings, and this wild type male flies. And in the same way, but in this cross he obtained only 62.8% parental types and 37.2% new types. Then Morgan concluded that the linkage is of two types. One is strong linkage, another is weak linkage. Strong linkage means parental combinations will be maximum, and weak linkage means these new combinations will be minimum. So here we have the very strong linkage, and here we have very weak linkage. That is why in the strong linkage we get only 98.7% parental types, whereas this weak linkage we got only 62.8% new types, parental types. So that is Morgan's cross. After studying the Morgan's cross, this student Chaithanya developed a technique known as this gene mapping. The principle of gene mapping was that if the crossing over recombination percentage is great, then the distance between genes is great. And if this recombination percentage is very low, that the genes are very closely packed. So according to the idea of Chaithanya, when the crossing over percentage, recombination percentage is maximum, the genes are separate, and when the crossing over percentage is minimum, the genes are very closely packed. By using this technique, by crossing over data, we can make the genes, we can map the genes on a chromosome. Now this is another modification that is polygenic inheritance. In polygenic inheritance, several genes are influencing single character. And the typical example is this skin color in man. Skin color in man, 3 dominant alleles are involved, and they produce maximum pigmentation, and the recessive alleles, and they produce only minimum pigmentation. These are known as contributory alleles. These are known as non-contributory alleles. And in the F2 of this polygenic inheritance, we get a bell-shaped curve. And these polygenic characters are easily influenced by environmental conditions. Okay. This pleiotropy means a single gene influencing more than one character. And the typical example is this phenylketonuria. Along the phenylketonuria, it is a single gene mutation. This through phenotypic aspect characterized by Mendel retardation, reduction in hair, skin pigmentation, etc. Phenylketonuria that we'll discuss in the Mendelian diseases. We'll be having, we can discuss it there. Next is sex determination. It was first this Henking he noted that there is a chromosomal difference in chromosome number between male and female in some of the insects, and we found that males are X0 and females are X6. And these males are lacking one chromosome, that is why it is written as 0. And whereas females have two such structures, and in male sperms, he named it as this as X body. X body. Later, this X body is represented as chromosome. And since it has some sexual mechanism, it is involved in determining the sex. This X chromosome is later on came to be known as this sex chromosome or X chromosome or sex chromosome. And in sex chromosome, there are three different methods like males can be males heterogametic, whereas females are homogametic. Females are homogametic. Male heterogametes can be of two types. They can be X0, as in the case of insects, and it can be XY, as in man and Drosophila. Man and Drosophila, it is XY. Whereas in both the cases, the females are homogametic, XX. And here, the males produce two types of gametes, one with X chromosome and one without any sex chromosome. That is why it is represented as 0. And in man, this X and Y in man is in man, this X chromosome is a long chromosome and Y chromosome is a small chromosome. Only this much portion is homozygous. Whereas this much portion, they do not have any Y chromosome. So whatever genes present on this X chromosome will be represented. This condition is known as hemizygous condition. Hemizygous condition. Due to the absence of this Y chromosome, the characters present on the S chromosome is expressing. So this is hemizygous condition. In Drosophila, S chromosome is a long chromosome and Y chromosome is a hook-shaped chromosome or a J-shaped chromosome. In both cases, females are homozygous. Then, if we make a cross, a normal man and a normal female, we get X, Y, X. Now, these two are XX, this is female, and this is male. So, in a normal man and woman cross, we get 1:1 ratio between male and female, and the only X chromosome that the male receives is from his mother. The father contributes only Y chromosome. The female receives two X chromosomes, one from the father and the other from the mother. And so in domestic chicken, we have another type of sex determining mechanism where this male is homogametic and female is heterogametic. So male homogametic and female is heterogametic, and males are ZZ and females are ZW. This is a modification of this males homogametic and females are heterogametic condition. And in sex determination mechanism, this heterogametic, it is the heterogametic individual, whether it is male or female, that will be determining the sex of the offspring. If it is in male, in this case, male is determining the sex of the offspring. If it is the female is determining, as in domestic fowl, domestic chicken, female is determining the sex of the offspring. This is pedigree. Pedigree means there are several diseases or particular characters which are inherited in a particular family only. So how these characters are inherited in that particular family, that we can study only by pedigree analysis. In pedigree analysis, we cannot make controlled mating experiments on human beings. So in plants and other animals, it is possible. So we have to trace the history of that particular character by studying the detailing the family history. And in this family, after that, after studying the family history, we draw a tree known as family tree, this pedigree tree. Here, male, female, affected in mating, consanguineous mating, like that. The next is genetic disorders. Genetic disorders can be of two types. One is Mendelian disorders and another is chromosomal disorders. Mendelian disorders and chromosomal disorders. Chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation of a single gene, and this can be transmitted on the same as in the case of Mendelian pattern of inheritance, and we can study only this by pedigree analysis. And most common examples are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, and thalassemia. Whereas these chromosomal disorders are due to this excess or decrease of one or two chromosomes. Okay, that we'll study later after this Mendelian disorders. And this Mendelian disorders, Mendelian disorders can be dominant or recessive. It is changes in the genes, so it can be dominant or recessive, or it can be autosomal or sex chromosomal on the sex chromosomes. And first, this is, we can study, that is hemophilia. Hemophilia, that is the inheritance is just like hemophilia and this color blindness are same. The female, so this is a normal female, and this is a normal man. This is a normal man, and this hemophiliac gene is a X chromosome recessive gene. X chromosome recessive gene. A female to be born to be a hemophiliac, a female to be born to be hemophiliac should be like this. In this case, the female should be either she should be minimum she should be heterozygous, and the father should be a hemophiliac. Then only there is a chance for this condition. So this is a very rare chance. And the first, it was noted in Queen Victoria, and she was a carrier. She was an heterozygous carrier for the disease. And this all sex-linked or X-linked inheritance, sex-linked or X-linked inheritance shows crisscross pattern of inheritance. Crisscross pattern of inheritance. Crisscross pattern means from grandparent to grandson through daughter. That is the inheritance pattern. It is not directly transferred from the father to the son. Father gives only Y chromosome to his son, and the mother is actually giving that disease genes to his her son. Okay. The same type of inheritance is also seen in the color blindness. Next is sickle cell anemia. In sickle cell anemia, there are three types of hemoglobin like that. This is normal hemoglobin, and this is sickle cell hemoglobin, and this is heterozygous condition. When these two heterozygous condition people, when they can produce a homozygous sickle cell anemic condition. In sickle cell anemic condition, the RBC, instead of they becoming round and normal, it becomes sickle shaped or crescent shaped RBC due to this some defects. The problem is caused by a gene mutation, single mutation in the gene, and single mutation in genes. And as we know, there are two alpha and beta parts in hemoglobin. Two alpha there and two beta chains are there. In this alpha, there is no problem, but in beta, on the sixth base, instead of this glutamic acid, the sixth amino acid is valine. So here we have valine, histidine, leucine, threonine, proline, glutamic acid, whereas in sickle cell anemic, we have valine, histidine, leucine, threonine, proline, valine, and glutamic acid. This valine caused the problem. And this is due to normal condition. This is the genes and this is the mRNA, whereas these genes, A and T, a single base mutation took place. So that mRNA is changed. GAG brings glutamic acid in the normal case, whereas in sickle cell anemic case, GUG gives only valine. So that causes the problem. So that is sickle cell anemic condition. Next is this phenylketonuria. It is an inborn error in inborn metabolism, and this is autosomal recessive condition. And this is due to the presence of phenyl. It cannot be, the individual cannot convert phenylalanine into tyrosine, that enzyme, phenylalanine phenylalanine, this hydroxylase enzyme. This enzyme is lacking, and so they cannot oxidize, they cannot convert phenylalanine into tyrosine. So that this phenylalanine or phenylalanine or sometimes phenyl pyruvic acid or sometimes phenyl lactic acid is accumulated in the brain and spinal cord, etc., and they will show mental retardation, etc., and it is excreted also through the urine. So this disease is known as phenylketonuria, inborn errors, that is due to this autosomal recessive condition. Next disease is this thalassemia. Thalassemia, also autosomal linked recessive blood group disease transmitted by parents to progeny when both the parents are heterozygous. And there are two types of thalassemia, alpha thalassemia and beta thalassemia. This alpha thalassemia means alpha globin chain will not be produced, and in beta thalassemia, beta globin chain will not be produced. So both these, only when both these are produced, a normal hemoglobin will be produced. So here the quantity of hemoglobin produced is less. Either alpha is not produced or either beta is not produced. And this alpha thalassemia, production of alpha globin chain is affected, and beta thalassemia, beta globin chain is affected. Alpha thalassemia is due to this mutation of two genes, HBA1, HBA2 on chromosome number 16, whereas beta thalassemia is caused by a single gene mutation, that is HBB on chromosome number 11. And thalassemia is different from this sickle cell anemia. Sickle cell anemia, the hemoglobin is produced, but the hemoglobin produced, this quality is different. Wrong type of hemoglobin is produced. Whereas in this thalassemia, the hemoglobin is not produced. So thalassemia is a quantitative problem, whereas sickle cell anemia is a qualitative problem. Next is this chromosomal disorders. Chromosomal disorders, this is due to this aneuploidy. Aneuploidy means increase or decrease of chromosome number. One or two chromosomes are excess or decrease, that is aneuploidy. And in the case of, there are three diseases we have to study. First is this Down syndrome. In Down syndrome, there is in Down syndrome, it is a genetic disease, presence of additional copy of chromosome and chromosome number 21, that is trisomy 21. And the individual baby will be like that with congenital heart diseases, broad flat face, and many loops on the creases, and he will be epicandal eye fold. Epicandal fold of eye fold. His eyelids will be very thick, and that is epicandal eye fold, and physical and psychomotor mental ability is very low. That is this 21st chromosome is represented 3 times, and these chromosomal defects can be studied only by karyotype analysis. By chromosomal studies, it can be studied, only studied, and it cannot be studied by pedigree analysis, because this disease is all of a sudden, the disease is appearing in a normal family, maybe due to some chromosomal defects or some other mutation. Okay, so that is this Down syndrome. Next is the Klinefelter syndrome. In Klinefelter syndrome, there is, instead of normal 46 chromosome, these individuals will have 47 chromosome. Usually the normal adults will have, normal individuals will have 44 autosomes, plus males will have 2 sex chromosomes, XY. Instead of this, in this particular case, there are 2 X and 1 Y chromosome. So the total number of the chromosome number is 47. This is known as Klinefelter syndrome, addition of a copy X chromosome resulting in the karyotype 47, that is XXY. Such a masculine development, but many female development like gynecomastia, and the individuals are sterile. And these individuals can be called as sex chromosome trisomy. Sex chromosome trisomy. And lastly, we have this Turner syndrome. Turner syndrome is seen in females. The females are sterile, and the females, instead of 46 chromosomes, they have only 45 chromosomes. For example, the individuals with the 44 plus X, and there is no second X chromosome is not represented. So we can put it as 0. So they have only 44 plus 1 X chromosome, that is only 45 chromosome. such individuals are showing this Turner syndrome, the females, and the females, their ovaries are rudimentary, and this can be called as sex chromosome monosomy condition. So that is a quick revision of this genetics chapter. This is comparatively a long chapter and very important chapter also. Okay, study well. Okay, thank you.